So today we have two ongoing project presentations that can assess their sustainability in future European calls to cancer fighting strategies. In first place, we want to introduce you BIOCELLPHE, a lab-on-chip for cancer diagnosis. Researchers will create a device for the identification of the tumor cells carried around the body in the blood circulation. This investigation can suppose a new era in medical diagnostics. The project it is coordinated by Vigo's University, Universidade de Vigo, and we have Gustavo Bodelón, researcher at Thimbio, its multidisciplinary research center, to talk about it. Mr. Bodelón, activate please your sound. Welcome, and whenever you want.
Well, hello, thank you for this kind introduction. I'm trying to put my presentation in bigger screen. Let me just give me a sec, please, if I can do it. Oh, sorry. Well, okay, well, I'm gonna get started like this. I don't know what's going on. Well, okay, so my name is Gustavo Bodelón González. I'm a molecular biologist working at the colloid chemistry group, which is located at Thimbio Research Center at the University of Vigo. So our project BIOCELLPHE was awarded with 3.6 million euros in this H2020 FET Open call. Okay. So the consortium is composed by 6 research groups, 2 small companies, and a clinical partner from 4 countries, Spain, Portugal, France, and Italy. So, the challenge in BIOCELLPHE is to generate a new technology for the profiling of cell surface proteins at the single cell level. So as a key driver for realizing personalized medicine. So to this aim, we gather experts in the fields of synthetic biology, nanotechnology, microfluidics, and artificial intelligence in order to fabricate a biosensor lab-on-a-chip device. So the aim, as I just mentioned, is to use this device for the profiling of surface proteins present in circulating tumor cells in the context of breast cancer, to be able to enable a new advanced diagnostic tool. So as you know, circulating tumor cells are tumoral cells that are released from primary tumors, and they navigate through the blood circulation system, and they can exit the blood circulation and enter and metastasize into other organs where they can grow into new tumors. So the detection and the identification of CTCs is through liquid biopsy is a new means, or is a means to diagnose metastasis. The problem is the detection. Okay, so as I said, CTCs are often present in very low numbers in the bloodstream of patients. So besides the enumeration of CTCs as a biomarker of metastasis, it is also important to perform a molecular characterization of the proteins, the protein contact that is present in these cells for personalized treatment. So in the surface of CTCs, there are a series of surface proteins that can be viewed as biomarkers. So the identification of this set of proteins is important for prognostic and therapy purposes. So our idea or vision in BIOCELLPHE is to engineer bacterial biosensors is to engineer a synthetic transmembrane signaling system, which is shown here. So we will engineer a number of proteins as sensors and also transducers that will, upon binding of the bacteria cell to the CTC surface, the bacteria will scan the surface of the tumor cells, and the sensors will bind to these cell surface proteins, and it will inform these transducers that are located in the inner membrane to trigger a signal in the cytoplasm of the bacterial cell that will end up producing this set of Raman reporters. These are specific molecules which can be detected by surface enhanced Raman scattering microscopy. So CERS is an analytical tool that uses plasmonic gold nanoparticles as optical enhancers to increase the inherently weak Raman signal of molecules when they are in close contact with the metal surface. So CERS is an extremely sensitive analytical tool that can achieve single molecule detection level, and it's also characterized by high multiplexing capabilities. So theoretically, it can detect up to 100 analytes simultaneously. So, so the idea is to fabricate, well, one of our collaborators has fabricated this microfluidic chip that can isolate CTCs from the peripheral blood of metastatic breast cancer patients, as you can see in this CTC, which is trapped in here, these microfluidic features. So our aim is to fabricate a similar device that contains this plasmonic nanostructure substrate that will detect the secretion of Raman active molecules when these sensors bind to the cell surface proteins of CTCs and trigger the production of these Raman active molecules, which can be detected on chip by CERS. So to conclude, so BIOCELLPHE's radical advancement will provide a solution for a yet unmet challenge, which is the synthetic transmembrane signaling in bacteria, which has not been shown to date. BIOCELLPHE engineered bacterial cell sensors will report the presence of protein biomarkers on the surface of breast cancer CTCs simultaneously with high sensitivity at the single cell level in a lab-on-a-chip device. So BIOCELLPHE disruptive technology for detection of tumor cells in bodily fluids can further pave the way for the early detection, prognosis, and therapy of cancer, as well as personalized medicine. I would like to finish my talk acknowledging BIOCELLPHE Consortium, the members of the College Chemistry Group, in particular Isabel Pastoriza Santos and Jorge Pérez Justa, and also Almudena Justo Martinez from FEUGA for her kind help at the beginning of the proposal. I would like to thank all of you for your attention. Please email me if you have any further questions, and please visit our webpage. I'm sorry for any inconvenience that these problems during the presentation may have caused to you. Thank you so much.
Thank you so much, Mr. Gustavo Bodelón. It is normal to have technical problems, but we apologize to the participants for our technical issues at the beginning of this presentation. Before you leave us, I would like to do it a question. Do you think the technology, this technology that you just explained to us, could be validated in other type of cancer, or do you think you could detect other type of diseases? I don't know, such as COVID, for example.
Well, yes. For example, for COVID, we'll have to engineer these outer membrane sensors that, upon binding to COVID, they will trigger the same signaling event, resulting in the synthesis of these Raman active molecules that can be detected by CERS. Yes, this is possible.
So in the future, it could be developed, something like that.
Yes, yes, yes.
Okay, thank you very much for sharing your knowledge with us today. So the other project we want to talk about is BCAST, which means Breast Cancer Stratification, a project which identifies a large number of germline variants that influence the risk of breast cancer. In combination, these variants can identify women at wide range of genetic risk, even in the absence of family history of breast cancer. To explain all the details, we are going to listen to Ángel Carracedo, researcher from University of Santiago. Mr. Carracedo, welcome.
Thank you so much, Alda. Welcome. I think I'm going to shift to Spanish, although it's almost the same
No
for
problem.
me. But anyway, it's bueno, ya no sé en qué estoy hablando, pero voy a describir muy brevemente.
I'm going to explain very briefly this project, which is called BIOCELLPHE, as Aida said. This project was funded by the Horizon 2020 program, which was supposed to finish last year, but is going to continue this year. The idea is understanding the determinants of the prognosis and the risk of breast cancer, of the subtypes of breast cancer and of breast cancer in general. The rationale behind it is very clear. Prevention is very important. In order to prevent breast cancer, we carry out mammographies with just selecting women through their age, but there are many other things that influence, like epidemiological characteristics and also very much the genetics. Almost 30% of variation that explain the appearance of breast cancer is genetic. This is something we get to know more and more. Why don't we apply these genetics factors? This is the origin of the project. This is something we have been working on for many years in a consortium which is called BCAC, Breast Cancer Association Consortium, where we have geneticists, pathologists, clinicals from many parts of the world working on a very similar idea to accumulate data that help us with this. We have had previous project financed before BCAC. In the last call, we applied two projects. One was Bridges and the other one was BCAST. Bridges is to see which variation we have in the germinal line that explains the risk of breast cancer. And BCAST is a variation, mostly somatic variations. The tumor, when it's produced, it has different effects on different people. It accumulates mutations in a different way. This is what we want to analyze here. This is the continuation of PANCAN risk, a project I coordinated, and the idea was predicting risk in cancer, also in Horizon 2020. That is a project that was finished a couple of years ago. The project's objectives are these: understanding the determinants of risk and prognosis of breast cancer, taking this to models that can be used in real life, and also trying to promote raising awareness on how this should be integrated in public policies to improve the prevention of breast cancer. We had 8 sections of work, one of coordination. In 2 of them, we had clinical data of the tumors and also histopathological data, genomic data, which is the pack that I was coordinating personally, and another package related to risk factors. Obviously, obesity, there are many, lack of exercise and so on. There's many environmental factors that affect the problem. All this we integrated, we created models for the prediction of risk, we validated them, having these models of prognosis and risk online, and then transfer them to the clinical practice. Our working group worked mostly on the work package 3, which is the panel of tumor mutations. It was really, really hard work. We had to extract the ADN of 10,000 tumors and of 10,000 healthy tissues from the same patients. We had to sequence gene panels, which is an enormous amount of information, and that was the bulk of our work. You can see this in the web of the consortium. If you click on the consortium web, if you write BICAS, you can see it. You can see all the work that was carried out there. This was combined with what was compiled in the second part devoted to pathology, and in the fourth working group, besides the risk factors, it included the mammography. So they took into account the different types of images. So we worked with a group from the University of Cambridge with risk modeling. This has been our work. All the information that has been generating is huge. This all has been integrated in prognosis models in order to identify patients with high risk, patients with moderate risk, patients with don't have any risk at all, in order to predict, be ahead of the problems. We have two tools that are at the disposal of all the health professionals in the world. We have CANRISC, the body theory model, and PREDICT. These two models are at the disposal of all doctors in the world. They are greatly used, particularly in the United States and in the United Kingdom. PREDICT is also used mostly in these two countries, but in many other countries in the world. We are carrying out work sessions, and people are using this in their clinical practice. In Spain, we have a pilot plan in Navarra, and we hope that this is soon implemented in Galicia too, and other plans are also being developed in Europe, and we hope that very, very soon we manage to improve the prediction of the breast cancer. This is the consortium, which is led by Machalka Slicks, and I'm learning every day from her. She's absolutely brilliant. She's leading marvelously this consortium, and this is a very complex consortium. It's absolutely wonderful being a part of this group. These are free groups, independent groups in this field in Europe. I hope this is continued in the future, in future projects. This is what I wanted to mention. Thank you very much.